he's not going to use it.
Okay.
So I forget her name. The other, she already took it and stowed it away.
I can bring this up just in case you want to use it
Okay.
for any reason.
Sounds good. You okay with people taking pictures? You okay with people taking pictures? Okay. And then, yeah, that's the only thing I needed. Yes. Okay. Yeah. Yeah. So let's see. Do you have a? Yeah, you have. Do you have a normal USB? If you just put it on there, and then if you don't mind, I'd have you talk first for I forget how long. Yeah, that's great. Yeah. Yeah, 10 minutes. Yeah. Oh no. Okay, so it's not. So that's not working. Okay. Let's see. I think we can switch you over. We can put on HDMI. If this is the easiest, especially if you just want to talk first. Plug into there. Okay.
You're able to just kind of bump that down, and then
Yeah.
I can just plug it in.
I think so. Right there.
Let's see what we're doing.
There we go.
So I'll wait to hop up until we're ready to go, or if we're tying up.
Yeah. Let me just double check, see if anybody's coming.
Yeah, for sure.
Yeah, no, I'll just give a quick,
Yeah.
you have to do a couple housekeeping things.
Perfect.
Jason? Sorry?
Yes.
Okay, from BD?
Mhm.
Okay. Okay, I think we'll get started now. So thank everyone for coming to the session where we're talking about implementing single-cell proteomics in a core facility and kind of the lessons that we've learned in our process of trying to do that. Just a few housekeeping notes they have to go over. So if you could please silence your phones. Photography of ABRF meeting sessions is permitted, and recordings of the program sessions will be available on the meeting website following this annual meeting. ABRF social media policy is that everything can be shared. If a speaker asks that it's not to be shared, please respect the speaker. Ben, you okay with sharing stuff on social media? Okay. Yeah, so feel free to share anything you want on social media. Hashtag for the event is ABRF 2026. We'll hold questions at the very end, so after both Ben and I talk, I have a couple of pre-prepared questions that we're going to talk about, but then anyone else can feel free to walk up to any of the microphones to ask us any questions. Download the ABRF app to view the full schedule, access Lightning Talk abstracts and posters, and connect with other attendees and sponsors, play the ABRF game, and create your personalized meeting schedule. All attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. You can also provide feedback for each session in the app. Look for the evaluate the session link within the session. But first, since it's a sponsored talk, we're going to have a brief talk from Jason from BD to lead things off.
All right, if everybody can hear me. Excellent. So my name is Jason Cove. I'm from BD Waters Biosciences now. I'll be talking to you super briefly at the beginning of this about flexible site-seq workflows on the Rhapsody and implementing them in single-cell protein profiling. So it gives you some storage options for flexibilities in these workflows. This is our Waters disclaimer side. BD's been purchased by Waters Biosciences. We're excited to be a part of them and join them, so you'll feel no changes, but you'll see some visible changes in the brand and everything else. All right, with that, I'll move into my talk. So multi-omic workflows across geographies for implementing for single-cell protein profiling. Storage options to this can be tremendously helpful because without this, having a multi-omics option or having storage options for multi-omics can create some challenges. If you have distributed collection, maybe these are sites that are happening from across different geographies or across the world overall, just having flexibility to be able to store these samples too at different time points as they've been collected, or to be able to batch process them. Maybe you're yourself or a core and you're receiving samples from many different people, or your particular customers want to be able to collect samples and are looking at reference from you. And then finally on this storage really supports operational efficiency too, just being able to batch process things as well. So noticing these challenges, Waters Biosciences really wanted to evaluate this and look if there was any sort of solutions we could come up with for giving a little bit of the storage flexibility. And that's really what we've developed the OmicsGuard Cryo Buffer for, and I'm going to show you a little bit of data throughout this. But really what this is giving you flexible single-cell storage options to effectively keep the sample in stasis and then be able to do either single-cell proteomic workflows on this later or various different single-cell assays. And we've tested this across a number of different assays on the Rhapsody portfolio. That doesn't mean that it's only compatible with these, just that these are the ones that we've been able to test because we have access to this. And it's a one key element of any kind of storage and collection model is how simple the workflow is going to be. The simpler it is, the more it's going to be easier for other people to use it, and any of your kind of clients or customers that may be interested in using it. And so for this particular workflow, we wanted to make it as simple as possible, and it is effectively just a buffer swap out. And so any sample, any media, if you're kind of cells or tissue or anything that comes into, it's really just about swapping that media out for the Omicscar cryo buffer, and then putting it into a slow cool and a Mr. Frosty application at all, and then it goes straight into the freezer for either 2 months in the minus 80C or up to 9 months, is what we validated now in cryopreservation storage. And so the first thing after, we've got a good workflow, we're trying to solve some of these problems in terms of, giving people more options for what to do with their cells and their samples afterwards. So we did a study with 2 separate samples in this, so about PBMCs in a particular tissue. So the first thing we wanted to look at was, in our kind of time course, when a sample is collected or when a sample is stored, are we preserving the viability across these different time points? And here these viability dyes are done with the BD Rhapsody scanner that it's just measuring the viability of cells that are put into the workflow. And so what you'll notice here is that really at these kind of across different time points with this, the kind of cell viability of fresh samples measured with PBMCs on the left-hand side with the blue. It's preserved really well across these different time points. And on the right-hand side in the orange, we have another kind of tissue maintaining the viability of that across 2 different replicates. And this is kind of how some of those studies were actually set up here too. So is that PBMCs stored for a bit and then run on the site-seq workflow with Beta Rhapsody, and then the human lung cancer tissue as well. The PBMCs were collected and kind of stored at the BD site, so we have a little bit more fresh data from doing this. We're not collecting any human lung cancer tissue at the BD site, thankfully. I don't think it would be very safe or recommended. So that was done with a collaborator. And so we'll have a little bit less fresh comparison data there because it was preserved and then sent to us to process. But for our PBMC samples, the first thing we wanted to do in our site-seq workflows, because gene expression is an important part of doing that as well as the protein expression data. So we wanted to look at the correlation between a fresh sample and the number of genes that are counted in each specific cell versus a storage time point in here. And we did this across 2 time points. So it was a 10-day time point and a 2-month time point. And as you can see here, the correlation, we've got a 0.99 for the 10-day and then a 0.97 for the 60-day. I don't think it's necessarily decreasing over time in that. I think that's just the sampling difference between the 2 of them. So we're seeing really high gene correlation across these samples with fresh and then in different storage timelines, which we're excited about. The next thing we wanted to look at was the clustering overlay from this. So in a given sample for fresh and frozen, are we seeing the same kind of distribution of cell types? Are they kind of overlaying? And you want the kind of the orange and the blue dots to really be right over top each other. And we're really seeing that here. We're extremely limited changes, giving us good confidence that the ways in which the cells are being analyzed and the data represented in those cells are reflective of the fresh, even when they're stored in the cryopreservation workflow. And on the right-hand side, you're seeing the overall UMAP and the cell typing from this. We're getting very nice tight clustering on this, just gives us confidence, we're not getting data that is losing its meaningfulness or its dimensionality in these workflows. The next thing we wanted to look at too, to convince ourselves that we are doing a good job at storing these and not biasing them in any way or any substantial way, is to look at the representativeness of the different cell type proportions in a buffer versus fresh, and then versus the frozen time points in here too. And so you see in the orange, that is our fresh representation or proportion of a sample that a given cell type was. So you want to see that maintained kind of throughout. And then we have those other 2 time points that I was talking about earlier. The light blue is the 10-day, and then the dark blue is the 2-month time point. And what you can see from in this particular figure here is that we're getting less than 10% variation for many of these different cell types across the storage times, which we're really excited about too. Boring is good in this particular case. We want it to be flat, and we want it to be nice across. Is no change is good change in this particular. Next, we wanted to look and make sure that our kind of site-seq or our antibody protein profiling data from this is preserved during storage. And so this is the same thing for these PBMC samples. So we looked at this after our gene expression was done. We needed to convince ourselves, is our antibody data and the sensitivity also looking robust when we store, so giving that flexibility in here. And what you can see is the bars are pretty much on top of each other there, which is great. These were sequenced to, you can see the sequencing depth of each of these samples here, and this storage sample that I'm reviewing, this is the 60-day sample, not the 10-day sample. We just didn't want to have all of those bars on top of each other for the figure. But you can see the long-term antibody data is preserved during storage in here, and you're getting really kind of equal data and sensitivity across these workflows, even with the storage time. Next, we wanted to review in this tissue workflow here the protein profiling sensitivity. With this, I took a few slides of data out here to keep the talk brief to make sure we can get to the kind of the bulk of things. But the next thing in this particular tissue for lung cancer, we also wanted to look at how is this protein data looking. Are we still getting sensitive data after a long storage time? These samples were stored for 9 months, these lung cancer tissue samples. And in this, in terms of the median molecules, that think about each kind of count of a given molecule. So you want to see this particularly high in these workflows just to make sure, okay, I'm going to get representation, good counts of these cells. And so there's not a reference in this particular one because we don't have that fresh because we're not collecting lung cancer samples at BD. But what we can see in this in terms of the about 10,000 or about 10,000 representation of molecules in there for kind of median or mean reads per cell. This is high sensitivity kind of counts and what we would really expect a tissue to kind of look like. So the storage is not kind of biasing or reducing the protein data that we've seen or had observed or expect with a sample. And then finally with this, I've got 2 more slides, I promise I'm brief. But we also wanted to look into, so we saw that we could have good quality protein profiling, protein sensitivity data, but then we also wanted to see after that, how does our clustering data look for a sample that was stored over 9 months in the kind of cryopreservation stasis conditions here. Because occasionally if the data might look good, but the clustering or all the kind of differences about the biology might kind of merge into each other if the storage wasn't, if the way that you're storing it isn't kind of keeping the representativeness of this. And so here what we're excited to see is all of the clustering data. We're getting really good clustering kind of separation and resolution of these different cell types that we would expect in the lung cancer tissue too. So we're really getting preservation in this of the tissue biology here, even after this 9-month cryopreservation storage. Finally, I'll kind of wrap up here and say we've demonstrated this across a couple different tissues. We have even more data that's going to be coming out in the very near future across a couple different sample types as well. I'll leave that to once it's actually announced. But if you want to go ahead and talk more or learn more about any of these solutions or anything on the Rhapsody, please come talk to us at the Waters Biosciences BD booth. I think we're booth number 214, 203. There we go. All right. Thank you, everybody, for your time. I appreciate it. I'll hand over the session.
Yeah. Okay. Thanks again, Jason and BD for sponsoring the session. So now we're going to get into the real meat and potatoes of the talk where really me and Ben are going to talk about our journey in trying to develop single-cell proteomics as a core facility service. Just as a brief outline, first I'm going to just give a brief introduction to the session, kind of the concept of what we're going to be talking about, an overview of single-cell proteomics and some of the breakthroughs within the mass spectrometry and proteomics field that allowed for this really recent rapid growth of single-cell proteomics. And then both myself and Ben are going to give brief 15, 20-minute talks on our own journeys through this, the mistakes we've made, our achievement, or like what we think about our current workflows. And then we're going to have a roundtable discussion between the two of us where I have several talking points that I've already prepared that we're going to talk about. And then with any remaining time, we're going to open it up to the audience. You guys can ask any questions from the two of us, or we can just have a general discussion between everyone here. So what are we trying to accomplish with this session right here? It's really, single-cell proteomics is hard. We don't have some of the benefits that single-cell RNA-seq has, where you can amplify your signal. Proteins are sticky. And so when you're working with such little material, any mistake or inefficiency you have in your method is really amplified in your end data output. And so all of us are really just too busy, and so we shouldn't be spending our precious time all making the same inefficiencies or same mistakes in our process. And a lot of the papers that I've been reading about this don't talk about mistakes that they made along the way, or whatever specific reagents they're using, or their bad data before they got good data. And so whenever you first read, like when I first read a single-cell proteomics paper, I thought, this method seems really easy. It seems really straightforward, so let's just go for it and see what happens. And of course, you'll see the data was not good. And so there's a lot of learning that we had to do along the way that the papers don't talk about. So I thought it'd be really helpful in this kind of setting to talk about some of those things that I know I learned throughout trying to do this over the past few years. Okay, so now we're just gonna briefly introduce single-cell proteomics. I was gonna have a couple poll everywhere slides, but I think Tim's session yesterday led us to believe that maybe that might not be a good idea, where we spent about 10 minutes trying to troubleshoot poll everywhere, so I just skipped those. The first question is, what is single-cell proteomics? What does everyone think it is? And really, in my mind, it's you performing proteomic profiling on a single cell with enough proteomic depth to be able to discern any kind of biological insight from distinct cell types or cell populations from a large collection of heterogeneous cells. And why do we want to perform it? I would imagine everyone's seen a figure similar to this, but just kind of get everyone on the same page. When you perform a bulk, any type of bulk analysis, you have your cluster of cells here, and in this figure they have 4 cell types distinguished by their different colors, and you perform mass spec on those. Your readout is, you can see here that there are different signatures or profiles from these 4 cell types, but they're really just all kind of covered up, and they're all averaging together. And so when you do data analysis, in this example of doing a PCA, you just get one blob of a PCA that represents the average of all of them. But then when you do a single cell and you sort out into single distinct cells, you can now get 4 very separate proteomic profiles from your mass spec data. And when you do a PCA, now you have 4 distinct cell populations. But what's made this viable more recently? And I think that there's really kind of 3 main sections where you have some instrumentation advances, sample prep advances, and then some data acquisition and analysis advances, which all combined have really helped to make this quite feasible. I'm just going to briefly go through each one. So instrumentation, some of these new mass spectrometers that have come out in recent years are very sensitive and really quite fast. So particularly the Thermos, Orbitrap, Astral, along with the entire Bruker timsTOF SCP lines, which includes the SCP, Ultra, Ultra 2, AIP. I didn't include it in here, but SciX's 7600 seems like it might show some promise to be able to do it. There's not much data on there, which is why I didn't include it, but I wanted to at least mention it. And then also we've had a lot of HPLC advances alongside of mass spec advances. So like the Vanquish Neo can allow for a really high pressure on it, so you can really load your samples a lot quicker. You can wash and equilibrate your columns a lot quicker, so your throughput is a lot faster with that. EvoSeps, the way that they design their pumps, allows you to preform your gradient, which is, again, leads to a little bit faster acquisition, and then it allows for faster cleaning and equilibration of your column again. And then we've had some really nice improvements in the actual chromatography. So like the ion optics columns, again, allow you to withstand greater back pressure and really high ionization efficiency, and Thermos micropack columns give really nice consistent chromatography between sample to sample. Then from sample prep, we've had a couple of nice sample prep methods that have come along in recent years. I think the real big one is the MPOP from the Scope 2 protocol from the Slavov lab. It's probably the most widespread one. Recently, there was the one-tip method that allows you to do all of your lysis and digestion within the EvoTip or on top of the EvoTip resin. And then the Car Lab has this Proteochip Evo 96 where you can do your lysis and digestion in a 96-well plate, but then you can invert that plate on top of your Evo tips and centrifuge your samples into the Evo tips without needing a pipette. And then we've gotten some nice methods to isolate and deposit the single cells. Try and true one is flow cytometry. But then Cell-in-One is a pretty nice unit for that, and that seems like a lot of papers are coming out. And then both Ben and I are, I think, both pretty solid constituents of TCAN Uno and really like how the Uno performs. And then we have our data acquisition, data analysis advances. And I think we all know that DIA has become, just in general, really popular over the past 10 or so years. And I think DIA in particular, the advantage that DIA proposed or has over DDA applies really well to single cell proteomics. So, you have your decreased sample to sample variation where you're going to have a ton of samples in single cell. Decreased missing values where we already have a lot now in single cell, but this helps to diminish that a little bit. And then you have, in general, your decreased bias towards your highly abundant proteins. Okay, so that was, like I said, a very brief overview. So now we're going to go into our speaker sections where I'm going to talk first, and then I'm going to hand it over to Ben. So just a little bit of background about my lab. So we're the Mass Spec Resource Lab at the Center of Advanced Resources in the University of Rochester. We have three main instruments where we have an Astral that's really just for bulk proteomics, phosphoproteomics, IPs, plasma proteomics, that we got in late 2023. And then in the summer of 2024, we got a timsTOF Ultra that the year afterwards upgraded to an Ultra 2. And this is really just for single cell proteomics and low input proteomics. And then our QE, we just have dedicated to targeted several targeted small molecule assays. And so I said that or at least for me, my single cell proteomics development is really a journey that's been happening over several years now. And this is just a highlight of each of the years of what we've accomplished in each of those years. So like I said, 2023 in October, we had our Astral installed. And once we had that installed, that was kind of when we thought, okay, maybe we can start to do all of these single cell assays, because that was a huge improvement for us over the Lumos. So that's when we did our first 250 picogram analysis on just a bulk digest. Got some good data from that. So we thought, okay, let's try and figure out how we want to perform our lysis and digestion. So we did those first experiments. But in 2024 is when we did our first analysis on actual single cells. That had some problems that I'll show in the next slide. Predominantly, we had a lot of contamination just from the buffers, so we spent a lot of time working on that issue. And then in, like I said, July, we had our Ultra installed, and then worked on just this for the rest of the year. And then 2025, we did a comparison between the Astral and the Ultra using the same single cell prep. And then in July of that year, we had our Ultra 2 upgraded. And then we worked with our genomics core to analyze K562 cells. And then just a few weeks ago, I collaborated with one of the researchers that we work a lot with, the Eric Wagner lab. And we did single cell proteomics on his actual model system, which is 293 T cells that are treated with this very specific drug. So I'm just going to go in a little bit more detail about those experiments I did along the way. So this was our very first attempt at true single cell proteomics, where what we did is we took HeLa cells and we grew them in media that contained heavy arginine and heavy lysine. And so the reason we wanted to do this is we wanted to get a kind of idea of the signal that we were going to be getting from any of the background proteins that would be present in the buffers. Because we know that if we got a heavy lipoprotein, that was going to be from a protein that was from the cell that we were trying to analyze. Because and as it synthesizes new proteins, it's going to incorporate these amino acids. And then any light protein would have been background contaminant noise from somewhere in the process. So this was our way of trying to tease out those differences. And you can see, and this is depicted just between any of the heavy proteins are going to be shown with this red whiskey plot, and then the light is going to be the blue. And so with just one HeLa cell, we were getting probably between 300 to 400 proteins on average, actual HeLa proteins, while we were getting close to 1,000 light label proteins. So that led us to believe, okay, we have a lot of background signal coming from something in our lysis buffer, our digestion buffer, from maybe the plates, the pipette tips. But that's also, we had a really low protein amount in our actual one cell, so we had a lot of work that we had to do. So I spent a lot of then early 2024 trying to tease out those differences, and so optimize some of the lysis and digestion conditions. And in March, we did another experiment of the same thing where we analyzed labeled HeLa cells, and we see that, okay, now we have 1,000 actual proteins from HeLa, while we have maybe 300 to 400 background proteins. So we kind of flip those numbers, which lets us believe that we're on the right track here. But we still, if we're only getting 1,000 HeLa proteins on an astral, we have a lot of work that we have to do still. So spent the rest of the year trying to just focus on removing these contaminants, optimizing lysis and digestion. And what I learned from this part here is that this is a continuing problem where the best way to do this, I think, is just you have your set stocks that are just for these experiments. You aliquot them into single-use containers, and you do all of that in a tissue culture hood, and then you only use one aliquot per reaction. July, we got our alternate evocept delivered, and we had to troubleshoot this for quite a bit and optimize that, so that was a lot of that year. We didn't know how to work with eye mobility. That was a big change for us, how to work with evocepts. And then in December, we decided to work with our genomics core that we have and perform a single-cell experiment with them, because they have been doing single-cell RNA-seq experiments for a really long time. So we thought that instead of trying to go into this blind, let's tap into that robust knowledge that the genomics core already has on working with single cells and see if we can apply any of that to our workflow. But as you can see, when we actually did an experiment with them, all of the single cells, the wells evaporated overnight. And I thought I'd load them in evotips anyway, see what we got. And of course, we got no IDs from anything. So that led me to believe, like, I tried this before, and this issue didn't happen. So we needed to come up with an optimized method that would need to be consistent from experiment to experiment, not prone to this variability that we were seeing. So we then further optimized all of these digestion conditions by ourselves, just using the TCAN Uno. So we spent a lot of time figuring out how to best validate these, and so we ended up digesting just at 500 nanoliters, and then we used this rapid digest mixture from Promega, which allows us to digest in only an hour, and we do that in a water bath, which both of those combined, I think, really helps to minimize the evaporation that we were seeing. And so we thought also, let's try to compare our Astral to our Ultra using the same prep. And so this middle bar here is the actual single cells, and anything with the blue outline, that's from the Astral, and anything the light blue is the Ultra. And we were seeing that with our Astral, we were getting probably close to 2,500 proteins on average between all the single cells, while our Ultra was about 1,200 or 1,300, which we weren't mad about at all, but we were thinking that maybe the Astral is really just that much better than the Ultra. But when we looked at our QCs, which is just Promega K562 digest that we diluted down to single cell amounts, which is 250 picograms, they were performing comparably to each other. So we thought that this wasn't indicative of, this difference is indicative of the performance of the actual instruments, but we thought maybe this could be us loading samples onto the evoceps, because this is just direct injection from the vanquish going into the plate and pulling out those peptides. So this led us to believe that maybe, okay, let's figure out how to best load the evotips more efficiently. And we had our Ultra 2 installed. Our app scientist, Andy Fan, who helped us with it, he suggested that, or he noticed that before, when you're loading less than 20 microliters onto an evotip, that for whatever reason, that's prone to variability when you actually load the evotips. So what he suggested was, before you load your sample, after you equilibrate your evotips and everything, you just add 20 microliters of solvent A into the evotip, and then you pick up your sample, your 2-3 microliters of sample, and put that into the evotip, trying to aim for that little 20 microliter reagent reservoir. And so doing that, now this is kind of just a summary slide of all the experiments that we've done over the years, with the red bar being our blank samples, which is just empty buffer with no cells, and then the blue is the actual single cells. Our most recent experiment that we did with the Wagner lab, we were getting close to 2,600 proteins on average between 96 replicates with very little background signal. And so each individual experiment looked like it wasn't very impressive. It's kind of disheartening each time. But now when you look at it, steady improvement over these past 3 years, it's really encouraging to see. And I just wanted to talk about this one a little bit more because this was our most, I was really most happy with these experiments because we also decided to compare the Astral to the Ultra 2. And our Astral performance is relatively the same as what we were seeing before, but now our Ultra 2, which I think is a combination of actually being upgraded to the Ultra 2 and learning more about how to properly load evo tips, our performance skyrocketed on the Ultra 2. There might be some room for improvement here. I haven't spent any time optimizing single cell methods on the Astral. I've been spending all of it just on the timsTOF because that's what we got that for. But then this plot on the right is a clustering of our results. And so just by doing some clustering, it got 3 independent clusters where you get this one cluster here that's all their DMSO treated cells. But then we get 2 clusters up here where these are all predominantly, few outliers, but these are all treated cells. So we have one large cluster here that's one cluster of their treated, and then another one here that's their other treated. So when they treat their cells with this drug, they're getting 2 distinct populations after 24 hours that are responding differently. And when we did Go analysis on them, we saw that this population has an upregulation in the proteasome, but this one has an downregulation in the ubiquitination system, which is almost a little counterintuitive. And when I talked with him about this, he said that when he's done bulk work for us in the past, he's seen some of his top hits when he does Go analysis is an upregulation in the proteasome and a downregulation in the ubiquitination system, which he said he doesn't care much about that part of the biology, but it always didn't make much sense to him that he would get those 2 systems that are very, very tightly linked to have opposite reactions. And so I think that this may be, by doing single cell, we were able to tease out, I think maybe, what was happening in his bulk analysis that he wasn't able to see. Okay, so just as I kind of want to go into what we're doing right now for single cell. And I've broken it up into 6 stages where you get your cell collection, where you collect your cells, and you have to isolate them into single cells, lyse and digest them, load them on evotips, acquire the data, and analyze the data. And there's really 2 ways that we're isolating our cells right now. It's either through flow sorting, through our flow cytometry core, or using a TCAN UNO. And whether we're using either of these, it changes very slightly our protocol. So if we're doing flow sorting, before we sort, we prepare a low bind 96-well plate by adding 200 nanoliters of lysis buffer, which is just 0.2% DDM with Promega's rapid digest buffer in each well, and we seal it. And then harvest cells, wash them with PBS, and resuspend them in PBS. Then we label them with DAPI so that we can exclude any dead cells, and then isolate and deposit single live cells into 96-well plate, and then immediately centrifuge it and transport it to our lab on ice. And then once it's in our lab, we add 200 nanoliters of digestion buffer, which is the same as this buffer up here, except we just add our rapid digest trypsin lyc, seal it, centrifuge it, and then incubate it at 70 C for a little over an hour in a water bath. And then once it's done incubating, centrifuge it, add your formic acid, and then incubate it at 4 C for 15 minutes. We're doing the UNO. It's, again, slightly different. Now when we're in the UNO, we're using a 384-well plate. When we tried doing a 96-well plate with their flow core, they were a little concerned about actually being able to accurately deposit that droplet into the very bottom of the 384 well. So we're just sticking with the 96 for them. But the UNO is really good at depositing the 384 well plates. You have to set up your, be a little careful when you set up your depositing scheme within that. We like to make sure that we have blanks in each batch that we're doing, a couple of carrier channels where we're just using 10 cells, and then you have to designate your junk wells. Harvest cells, wash them with PBS, and then resuspend them to 200 cells per microliter in PBS, and pass them through a 35 micron filter before you deposit them onto your dispense head, dispense the cells. And then we just pretty much do the same thing except to deposit our combined lysis and digestion buffer. We put them into the D1SF cassette, which then deposits 200 nanoliters of that into each well over like a minute. And then again, we just follow the same protocol as before. And then we load our EVO tips according to their instructions. And like I mentioned earlier, before we actually add the sample, we put 20 microliters of formic acid into the tips, and then we add the samples to the tips, trying to get it into that droplet. And then to acquire the data, we're doing all of this in using the Ultra 2, so we prepare our samples in HiStar. Just a little bit of details here. We analyze all of our single cells together, so we have them in a giant group, and then HiStar has this really nice function built in where you can just highlight as many cells as you want, and it'll shuffle your highlighted cells. So we just shuffle all of our single cells to remove any batch effects from the order of your plate position. And then we run all of our 10-cell carriers together, and we run our buffer blanks at the very end. For HPLC, we're using EvoSep1 with Whisper Zoom 80 with this Ion Optics 5x75 or Rapid, which is pretty much what you have to use for that. Mass spec, we're using the Ultra 2, using this DIA passive method that Bruker gave to us that the Bremen lab optimized. And I have the exact details on that on another slide I can share if you want. And then we search using Diant 2.3.2, which is the most recent version. And then analyze the search results with this SCP R package, and then just follow what they say in the SCP planar tutorials, which the links for both of those are right here. And I don't know if you guys want to, I won't go through all of these, but these are the materials that we're currently using. I think these plates are really important. One thing that I think is really important, or at least for us, was that we were using a regular DDM, and I noticed that that was where a lot of source of our contamination was coming in, was just the regular DDM. So once we switched to Thermo's specifically high purity DDM, that removed a lot of our contaminant signals. And then when we first got the Ultra, we were getting a ton of PEG background, which we think might just be related to loading on the EvoSep, where you're concentrating all of that PEG onto the tip. But all of that was removed once we used these Lycrosolve hypergrade formic acid solvents. So if you're using EvoSep with a timsTOF, you probably would want to make sure you get these solvents there. I can skip this for when Ben and I talk later. So just overall, some lessons that I learned when trying to implement single cell. First, achieving some of these crazy numbers that some labs are reporting in their papers is almost impossible, especially for a core lab where they're getting 4,000 or 6,000 protein IDs from a single cell. It's not, I don't think it's very realistic for when we're trying to run a ton of samples. They're able to run 30, 40 SPD methods using a cell-in-one and these very highly specialized equipment that's really expensive and going to take a lot of time, where we just don't have that luxury. Like I said in the very beginning, there's a little room for error with single-cell proteomics, so every little step needs very careful optimization, which takes a lot of time, and it's going to be a lot of quote-unquote failure. Like people say failure is worthwhile, but it sucks to have an experiment that doesn't work well, but you might get that a lot. Background signal must be carefully managed and controlled for. And then I think it's really important to speak early and frequently with others who have extensive experience, not just with single-cell proteomics, but any single-cell technique. There's a lot of parts of SCP that is applicable from other techniques. How they isolate cells, how they analyze their data, I think is a really important thing that we should talk with them about. I'm going to skip that part. And then some of our biggest challenges that we're trying to go through right now is we're trying to really get these sample protocols to be robust across not just the current cell culture samples that we're analyzing, but through a wide variety of types of samples, which we haven't currently done yet. And this is an important one, is determining the appropriate sample size for someone's experiment. These single-cell RNA-seqs, they're doing 10,000, 20,000 cells per sample. That's going to be, until we get higher level multiplexing or something, that's going to be really hard for us to achieve, especially for a core where we're going to be charging these out. Data analysis is going to be tricky, trying to figure out the best way to do that in a repeatable fashion. And then trying to communicate the benefits of doing single-cell proteomics over bulk proteomics, over bulk RNA-seq, or bulk single-cell RNA-seq. You know, this is a problem that, you know, proteomics fields in general has been having for a long time, but it's just going to be, I think, more important to talk about now. Just some brief acknowledgements of everything from the our mass spec core, the flow cytometry core that helped us a lot, the genomics core, grad student from the Gamagami lab, and then the Wagner lab and Thermo and Bruker for their support with their instruments. And with that, I'm going to move on to Ben.
That was a great intro. Thank you. Wow. Okay, there are more people here than in last year's Las Vegas. Okay, so quick talk outline. This is almost a victory lap for me. I think at ABRF Las Vegas, we kind of had a session that I set up, and Justin and I were like, wait, you're not gonna believe this. We're gonna try to do single-cell proteomics in a core facility. And so now I'm happy to say that the Pitt Health Sciences Mass Spectrometry Facility offers single-cell proteomics. We have paying customers, things are working, and that's, so victory, right? This is working. I'm gonna talk about my background in SCP because Kyle asked me to. Then I'm gonna go into our standard workflow. We only have one workflow going in the core. If you followed any of my work in the past at Hopkins, I was doing multiplex single cell proteomics, but it's just really easy with the label-free stuff to go and do that instead. It's more reproducible. There's less variables at the end. It's easier for the bioinformaticians. But if somebody comes to me and says, "Hey, we've got a 30,000 cell project that we will pay you to do. We'll pay the external corporate rate to the core. I will break out the multiplex proteomics and we will do TMT. And that cost will go down and it will totally work. We're just not a reason to do it yet. I'm talking about what's going well and what's not going well. My personal history with single cell proteomics, I saw this preprint in April of 2017. It was the first version. It was pretty bad. I got really, really excited about it. It was the first single cell proteomics. I was actually traveling around with the vice president of thermo marketing, and we spent a long time talking about how we could, what we could leverage this inside thermo to make this into something really, really valuable. He decided not to do it, and really they kind of lost the game on that. It was really, really exciting. This preprint Kyle showed, it wasn't actually published until 2021. It needed a lot of work to get there. But by early 2018, I was working for a big evil company, and I was doing single cell proteomics based on their techniques, using their instrumentation, everything verbatim that way. In 2020, I got the chance to leave a big, beautiful building that I had designed myself, and 80% of my salary behind, and go and join academia again as a non-tenure track faculty member to pursue this because I was still very, very excited about single cell proteomics. And we talk about, Kyle got into why to do this. I've got a review that we'll get the edits done on, but it's like, why would you do single cell proteomics, right? And so Kyle mentioned, complex cellular environments, and just a beautiful example that he described of where the proteomics it didn't make sense, and they solved it with single cell proteomics. Here, anything that we're looking at is actually a complex cellular environment, right? We got to break these down into cells. My personal research, we're currently very interested in a cell type called cholangiocytes, which I did not know existed until just recently. It's like 5% of the cells in the liver by numbers, and by mass it's much, much lower because they're very small cells. Those are rare cells in a population. I need to get those out and do single cell to get there. Most of my work at Hopkins was looking at, hey, even in cells in culture that have been growing since the 1970s, you add a drug, sometimes you will get these multiple responses to the drugs themselves, right? There's multiple adaptation phenotypes. But you can argue, well, that's all single cell technologies, right? They can all do this stuff, right? And the real punch, right, is that nothing out there can give you any input into protein activity that's driven by post-translational modifications except for this technique, right? Unless you're actually doing single cell proteomics. You can target some stuff with antibodies, but you're going to get 10 or 15, right? Maybe on a cytoph you can get 50 targeted PTMs, right, with antibodies, and not, but by mass spectrometry, you can do this very easily. Terminally differentiated cells are also very important. I like the red blood cell example, right? They don't even have a nucleus. They're not doing a whole lot of transcription and stuff. Like anytime they respond to stress, it's at a protein level. And something that's really, really interesting is the low cell input diagnostics. There's a whole generation of people very excited about single-cell-seq, and they were like, okay, well, we'll punch a hole in someone's pancreas and we can get 500 cells out, right? And we know what single-cell-seq looks like when you only have 500 cells, right? When you have 1 million cells, you can start to see these things. I will cancel my schedule. I will lie to grad students about where I am if you want to talk about low cell input diagnostics using single-cell proteomics. Right, we'll do that. Our workflow is, Kyle went through this. We're mostly using FACS. I like the TCAN Uno, but really it's just a viability marker. It works for what I'm currently doing. So you just use some sort of whatever, right? I don't know how any of this works, and we'll get back to that in a second. But I need those single cells to land in the bottom of the well of something all by themselves, and they have to be intact, right? So I don't care about a cell that has this ruptured cell membrane and it's floating around in the media, because that's not the proteins I'm interested in studying. Depending on who you are, you add lysis buffer and trypsin, or you do before or after. It doesn't seem to make a lot of differences. Because I'm concerned about post-translational modifications, my cells fall directly into pure acetonitrile, which I assume halts all cellular processes at the second that the cells hit that buffer, and that's how we work up. The trypsin cuts the proteins in nice little sizes for sequencing. Good job, BioRender. But you might be able to see in there, the peptides have been chopped up. They just don't fit in the well. Ooh, did that change the screen? Ooh, okay. All right, we're back. Okay, and then really, I get the best pricing on instruments that are shaped like this, but you can use instruments that are shaped like other ones, and Kyle has the Astral and the Ultra as well, but at the end of the day, any modern mass spectrometer that has ion accumulation can do single-cell proteomics if you are patient enough, right? You can accumulate the ions and accumulate. Now, on a retired instrument like a Q Exactive, labeled proteomics, you might get 4 cells a day, right? Maybe 8 cells a day, right? Is that a viable way of doing things? A lot of these instruments, they're not actually more sensitive, they're just more sensitive at speed, right? Our standard workflow, I'm just gonna blow through, it's basically what Kyle's doing. I get out of surgery, I get hepatocytes from patients that, they're for whatever reason, they're in surgery, we get parts of their livers. The TCAN Uno is perfect because that's all hepatocytes. We lyse that, use an EvoPacep1 system. Right now, we get 40 cells per day, and this is the core workflow that's validated. Yes, that is only 14,600 cells per year on an instrument that lists at $1.4 million, right? Now, so in January, I did have one of the final papers from Hopkins trickle out where we were doing multiplexing, got solid data at 700 cells per day. That's it's slipped down to Comms Bio. We use a timsTOF Ultra 2, same as Kyle. We have a online Diane 1.9 auto QC. It runs on every sample. We can just log in remotely and see, how is each cell in isolation running? We recently, thanks to brilliant work by my lab, we now have internal quality control. They've titrated the Biognosis IRTs. They're like 100 attomole in each cell when they're resuspended. We use 200 picograms of K562 for a quality assurance, intrabatch, and again, they're randomly inserted. The instrument method is saved as read-only because mass spectrometrists love to tinker with stuff, so they can't. And our final analysis is the Spectronaut 2.0 and SCP-Viz, which is hosted on the PIT HPC. What's going well is that I think we've got a viable cost structure for SCP. I spent a lot of time working on this, really, really popular preprint with everyone except for peer reviewers, where Amanda and I went through and looked at every single cell proteomics paper, and we worked out how much it costs them per cell to do that study. These numbers do change as you go across the board, but the very first single cell proteomics study that was done in bulk was about $1 a cell. Right? That's what it would cost me to reproduce that workflow. If you start to go to label free, you see that some of these numbers continue to climb and climb and climb. And oh man, viewers do not like this one. But just to be clear, this is just the instrument and reagent alone. There's no salary support, there's no electricity, there is no building, there is nothing happening here except for the depreciation of the instrument you just purchased, the amount of time it takes to run that cell, your service plans, and so forth. But you can range from a 50-cent per cell workflow on an older instrument that's multiplex to some of these newer things where with no person running it and no electricity, you're still at $30 per cell just in pure overhead. So we spent a lot of time thinking about this stuff. So again, I said viable workflow, right? What does viable mean? So just US Hupo, I told someone like, hey, internal, I think we're going to be able to get to $50 per cell. And he did this thing and just dropped a glass of wine. And he did not take my card. And but so completely mixed reviews. And so internally, we showed I showed this to a group at Pitt, and they were like, "Well, okay, well we're doing deep single cell seq, and this is less expensive if we can get." And we're basically in that range where we're coming in. Some of these like shallow single cell seq, where you send it to a company and you're getting 800 transcripts per cell. And you're really scaling it, you can get those numbers down, but it's not that crazy. But it all depends, right? And multiplexing, of course, brings that cost per cell way, way down. You can cut it, a 20,000 cell study will cost the same as a 2,000 cell study if you multiplex it, right? As you get kind of at the end. I made up those numbers, it's probably not quite close. The other things that are going well is our data is great. We've done a lot of cell types. I've been at Pitt for just over a year. Got 5,000 or so injections on my instrument. Everything's been going really, really well. My own personal research, hepatocytes taken right out of people from surgery. We're getting 2,500 proteins per cell with good quan all day from these workflows. We've got a couple papers in preparation. We collaborate with the Drug Discovery Institute, where they do all this organ on a chip stuff. They break open the organs on a chip. We take the cells out, dissociate, and we're getting that as well. Some small cancer cells, HepG2, we're getting close to 2,000 proteins per cell. Big cancer cells, polynucleated, we have some stuff that's 5,000. All this stuff is on public repositories. It's all massive. You can just go massive and just type Orsburn. It's spelled funny though. Mouse neurons we're working with, we're getting these out intact, 1,500, 3,000 proteins per cell. Now this is where it gets tough, like the tiny cells that have small protein content. This is where it gets challenging. I was collaborating with an immunologist, and some of the little cells were coming out. They were like sub 10 micron cells, like maybe sub 7 micron. We're getting 300, 400 proteins with the exact same workflow. Some of the bigger ones, they're doing okay, but it really is how much protein is in that cell. What's not going well is that every conversation, basically, I've been doing this 6 years, for real, full time, has ended with when I ask, how are you going to isolate your single cells, right? Because, because I'm not doing it. So go back to magic comment, I don't know how, right? And so this is the question, how do you get a single cell into the bottom of the well of a plate? I don't care how you do it, but you have to do it. And that ends a lot of conversations, because maybe that's still the most difficult part. Just this month, maybe the last 2 weeks, I have gotten 2 requests from people who have wanted to do single-cell proteomics on post-mortem human brains, right? And my understanding of the human brain being mostly composed of lipids is if you freeze that brain, then the cell membranes are no longer viable things, right? And so they can respond that, well, we do single-cell sequencing, and it's like, well, yes, you do single nuclei sequencing, right? Because the nuclear membranes are intact. Maybe that can't be done. But I guess at a certain point they could just start. We can start doing some method development, but I'm pretty sure you can't do that because that cell membrane is no longer intact. Kyle mentioned this. We've got the literature is distorting expectations. Last year, before I realized that they record this entire thing and then put it online, this preprint had come out. I called these authors a bunch of assholes, and they did change it when the paper was accepted, because they said in the title that they were getting 5,000 proteins per cell. They were not getting 5,000 proteins per cell. I've analyzed every data file in that. It still made the abstract. I was removed as a peer reviewer. I'm no longer listed. And so, maybe they got 3,000 proteins per cell. And so there's this buzz, like, hey, buzzword. I could get 40,000 proteins per cell. Nobody's checking it. This thing just came out in January in Nature Biotechnology. I just finished reanalyzing all of their data this weekend. They're getting 418.6 protein per cell and over 90% missing values in this study. All I see is batch 1, 2, and 3 in analysis of their data. Actually, 1, 2, and 3. So quick shameless plug. How do you reanalyze all these data? Because it's pretty clear that the peer reviewers aren't doing that, and I can't be the reviewer on every single cell proteomic paper now. There are just too many of them at this point. So we have this up on the PIT HPC. I'm going to show you some the stuff that's going in the publication version, but you can go there. It's rocket fast because, man, PIT has an amazing HPC. It should be like a recruitment thing, like, no, for real. We actually know how to do high performance computing here. You can do that. You can just Google it. It's online. You can run it. It accepts data from any proteomics software tool generated by any instrument, labeled, label-free. It doesn't matter. I'm going to show you 1.08, which isn't up on the server yet, but in general, this is 4 years of development on this, no coding required. And so Kyle's using the R package SC planar. I'm not smart enough to do that, but I am smart enough to take all the cool stuff that they were really happy about in SC planar and make someone else put that into a push button workflow, and that's what this does. Yeah, and we could do protein, peptide, or PTMs and visualize them all just right here. You can also run it locally. It's up on GitHub. And so it allows the discovery and parsing of weird cells. So here I had some cells over in that group where that little arrow is that didn't look like the other cancer cells from its group. And so you just circle them, and then I renamed them weird H358 cells. And then I can just do a quick clustering and say, okay, what's the difference between those? So part of this is, like, one, this makes it much easier for us to analyze our data. I format the data for customers into the format, give them the link, have them loaded up, and then they can see it. And also it allows me to very rapidly go back to data that's being published or has been published or in peer review, and I can just reanalyze and check their data real fast. Acknowledgements, thank you to NIH, at Pittsburgh for the amazing startup funds. My current lab's on the right, my lab, part of my lab from Hopkins is on the left, lots of external collaborators. My conclusions, so single cell proteomics has real world applications. We're getting requests for them, things that actually make sense. It's not just a bunch of nerds at ASMS just being like, how many proteins per cell can I get? And that's what the field felt like for the last 4 years. It can be implemented in a core facility. Kyle's got it running. Justin Wally, who presented at SEP or at ABRF last year, his core just went live. Einstein will be 4th. It's coming online this summer. I don't know how Simoni's going to do it. He's also becoming a father here in a couple months. So there will be 4 university core facilities probably by the end of the year that offer this as a service. It's still a new field. It's not without challenges. And then, a shameless plug, I've developed some tools that hopefully make the informatics less burdensome for everyone. So I think that was all I had. Yep, nope, just backup slides. All right, next section. Are we sitting over here?
Yeah. I think I was going to have prepared questions first, but I think it's going to be more useful if people first want to ask their questions, and then if we have any time afterwards, we can kind of fill in any time with those questions.
Yeah.
That one should be on, if you want.
Anyone have questions? I'll bring you a microphone. Oh, good. Jeez. Okay. I'll hold it.
Yeah.
Thanks for the talk. I was unaware that there's only 4 facilities offering single-cell proteomics. It felt like everybody was doing it, so that's good to know that I'm not as behind the 8 ball as I thought I was.
Yeah.
So I recently went to a webinar from Thermo Fisher, and they presented some information from a lab that's doing single-cell proteomics with the Astral, and they actually developed a 2-column system where they're going back and forth between 2 columns to improve their proteomics per day, and they were only doing 5 to 10 minute gradients. How long are your guys' gradients, and have you ever thought about doing the 2-column system?
You want this one?
Yeah, I can. So I'm using, so Evosep you can only use their predefined gradients.
Okay.
And so I'm using their Whisper Zoom 80 SPD, which is, I want to say it's 14 and a half minutes. And unfortunately, with the 2-column design, I think Thermo might be starting to sell them commercially. I know Chris might be able to. That may be a bigger problem with the Bruker instruments, just because the way that they have it designed is the emitter has to be like directly next to the IT, essentially their equivalent of the ITI. And so I don't know how you would be able to do a 2-column system with that. Someone's probably thinking about it.
Yeah, so Jenny, there's 2 competing technologies, right? So Jenny Van Eyck's got hers, the dual trap single column DTSC. It's 15.5 minutes run to run. It runs, if you buy you have an RSLC Nano and you contact Simeon Creamer, he will send you the whole walkthrough on how to set that up. I did get one of those secondhand for like 30 grand. And so thinking about how I can scale up my workflow, versus it didn't come with a good auto sampler. So, but am thinking about that. Ryan Kelly is at 6 minutes a run by using 2 traps and 2 columns. I went to visit him at BYU, and the LC has like glass capillaries like sticking out all over the place.
That's who presented it.
Okay, yeah, and so
they 3D printed their own stuff. So
it looks like something I could never set up in a million years.
Yeah,
and if I was like not paying attention, I would break all the glass that's hanging off of it. I think it's coming, and I think Thermo loves Ryan, and so I would suspect that they will develop that. But at this point, the EvoStep's just really fast.
Sorry, one more question. The Cellin one, I got a quote from them, and I put it back in the email, never to look at again because I'm never going to be able to afford that. But I'm just curious. They were kind of saying that the Tekken Uno drop or whatever it's called had problems with clogging. Have you guys ever seen that? If you could
Ben and I were actually just talking about that because I ran into that a few times. It does definitely help to first run it. That's why I mentioned it earlier, to run everything in the 35 micron filter beforehand. That definitely helps at least a little bit.
Yeah, I was curious about that because I wasn't sure why you'd want to use. Is that your single cells are going through 35 micron or the whole bulk?
So you have your cell suspension, and you just pass that through. Like we have like the cap, the tube cap. Filter, we just pass it through just to get rid of any of the clumps in there. So it's really then like a single cell suspension.
Okay.
Ben was talking, you can talk a little bit more about this.
So we, especially with some of the patient samples, when they come in and their viability is low, some of the liver will polynucleate, some of those things will clog cells. But when you clog the TCAN Uno, you're out $45, right? You toss this capillary, right? And so my Cell M1 at Hopkins was $350,000.
Yes,
and so I could just like spend the rest of the year just throwing TCAN Uno cartridges away. Plus, if you look at the capacity of the Cell M1, it's really low. I know that they work on this a lot, and they offered me a bribe to stop saying bad things about them, but it's not that like I didn't hate the instrument. It's just if I was trying to do a workflow that makes money, and if you look at, if you go to the single cell economics paper, if you look at the instruments where every cell before labor was over $20, they use that instrument because all of a sudden you've got this $300,000 instrument that costs $20,000 a year in service and has expensive reagents that now makes it just almost, makes it completely unviable.
Well, it seemed neat because you could do imaging of your cells as you put them into the wells.
Yes. And
that was like cool, but I was like, I don't know if anybody who's going to really care. So.
Oh yeah, no. There's another company bringing something that does microscope-based imaging to market, and it should be ready before the end of the year. I'm hoping to have it soon, and it's maybe a quarter of the price and 5 times faster.
All right. Thank you very much for doing this talk.
Thank you.
Hi there. I wanted to ask the question with the technology evolving and now only 4 by the end of the year being out there as a core facility option. How do you work or convince your faculty that this is something that they really need to do compared to other single cell techniques or other options out there available to them?
You ask them to validate their single cell seq findings at a protein level. And when they come back with like they're getting 30% validation, then you're like, okay, well, no, then come back. Let's do something that actually reflects the protein level. That was a joke. I'm sorry, I was being sarcastic. But yeah, really, how many single cell seq data sets have resulted in a diagnostic, right? It's like, oh, it points out a pattern, but it's like, okay, well, but what is that pattern? What makes those cells special? I think that people are starting to get tired of spending lots and lots of money on seq and then finding out that, well, that protein isn't actually reflective. But
do you have like workshops or seminars on campus that help people understand why it's important?
It's a good idea. We should.
We are like trying to start that with bulk proteomics. It's a time-consuming thing, and we're limited staff, so it's like we're trying to devote whatever time we can to that. But it's hard to try and convince people that there are benefits to not RNA-seq in general. That's why I mentioned it earlier, that we're having a hard enough time on bulk analyses. I feel like we are gonna have a harder time with single cell in general too, especially since we're gonna get lower numbers. But I think we have to figure out how to price it competitively too. That's why I was trying to talk with some of the faculty that do single cell RNA-seq. He was like, they charge us $10K to do 10,000 cells. So if you can, whatever number of cells you need to perform an analysis, probably try and price it somewhere around there or less, and maybe you'll get people at that point.
Thank you.
And even these papers that I pointed out that I'm not a fan of, they're doing great advertising for us. This Nature Neuroscience paper, it got me 3-4 phone calls. I despise that paper. The quantitative accuracy is awful in it, but man, what great advertisement it has been. Like all these people in neuroscience and psychiatry, and there's huge things in Pittsburgh on this. They've started contacting me because they saw that paper. It's like, well, wait till you see what we can really do, right? So.
Okay, so my question is similar to your question. I have a group, and I'm spatial, single cell, that kind of tech, no mass spec, right? And they wanted to just do site-seq. And I said, oh my gosh, you know how expensive this is going to be just to do site-seq. So maybe you should do something else. It's a clinical project, right? They want to just look at protein. Well, this paper did it exactly like this. Okay, fine. So we're going to do a small little project, right, where we do RNA and protein, and they have no idea what's going to happen next. I don't have this kind of resource on campus, but this is exactly what I think they should do, right? And I even tried to dissuade them from using my core, right? So how would you recommend for those of us that don't have a single cell proteomics core, what would be the next best step here to get people to do what they really should be doing?
We tried something called TotalSeq, which is essentially antibody, right, with barcodes on them.
Yeah, that's exactly what we're going to use.
Okay, got it.
Yeah.
Yeah, and so we tried that. We don't have any publishable data from it because that kit was expensive, and it works apparently very, very well in Gila, and that's what they give you all the protocols based on. And again, this was maybe a year or two ago. Well, it's before I got to Pitt, so it's 2 years ago. And trying to get it to work on mouse brains, what were dissociated, it just didn't. Like, we didn't have the protocols, we didn't have the support, it didn't work. But I think that those things must be getting better, and so then that's kind of like your entry into single cell proteomics, maybe.
Yeah, but I think what I'm talking about is that once they get their data, right? Because I'm confident we can do all of this.
Oh, okay. Sorry.
Then the next step, would they go with your kind of an approach, or should they make a flow panel, et cetera, because they are thinking they're just going to continually do single cell experiments with me. And what I'm explaining is it doesn't make sense for just the protein aspect to do a 10x capture and then not use the RNA library, see what I'm asking? And I think especially with clinicians, they don't understand. And if I don't have those resources on campus, I guess my best option is to say you should create a flow panel.
Okay.
Do you agree?
Yeah.
think that's going to be a case by case, because I know that's something that we're talking about with Roswell, and how much like Spencer is going to be involved in talking about the research, her researcher's data with the researcher, how much we're going to be involved with developing the experiment with the researcher. I think it depends on how the core is set up, like the expertise that the home core has, and then the where you're sending the samples to, how much free time you have to actually sit down with each researcher as well about all these extra projects.
Yeah, I think we're uniquely positioned because I'm a hybrid. I not only do biology, but I also do bioinformatics. And most of the projects I'm sending over to Kyle, I'm already intimately involved in. And so it's a lot easier for me to facilitate those conversations with the investigators. But like Kyle said, it probably is going to come down to a case-by-case basis. I know one set of investigators I sent over to them, it was just kind of a cold call from an institute in New York City who heard that we had some capabilities. And I can facilitate that conversation as well, just because I wasn't familiar with them or their project. So yeah, it's definitely case by case. But if it's investigators from your own institute where you've kind of been intimately involved, you've done some other assays with them, you know kind of what the next step should be, then it might be worthwhile to set up this type of interaction.
Yeah, and if you want to talk, I'm down for consortium. Sound like a great idea.
Oh, so my question is for Kyle. You had mentioned that for your SCP optimizations, you use the combination of timsTOF Ultra and EvoSep. And so do you think the same combination, do you think you can get the similar numbers with Orbitrap Astral if you combine Orbitrap Astral with EvoSep, or do you think optimizations just with direct inject are going to get you those numbers?
Yeah, I think that if I were to put either HPLC in front of either instrument, I feel like they would be similar.
But do you think coupling the instrument, the mass spec instrument with EvoSep versus Vanquish gives you a more robust setup, one versus the other?
I will say that so we are looking to trade in our EvoSep for a Vanquish. We don't like, part of it is, I think that we are, no matter what we do, we're going to lose sample from pipetting from the plates to the EvoTips. That's going to happen no matter what. We can get around ways to improve that a little bit, but it's going to happen. So ideally, if we could direct inject for every single sample, that would be ideal. And also, there's the added cost of the EvoTips. Each tip is like $3.50 to $4, depending on how much you buy at a time.
Yeah.
And so that's just doing a 96 well plate. That's essentially $400 in extra reagents. So that is.
So from the point of from the point of using a more pragmatic approach for having the ability of doing SCP in a core, do you think one should, or a core should have different instruments dedicated for non-SCP workflows versus SCP?
I think if you can swing it, you should. That's difficult though.
Yeah.
We're a very unique case where we submitted an S10 for an HT too, and then our Lumos died before we got our score back for the S10. So we were like, we need an instrument, and then 2 days after we submitted our S10, Thermo was kind enough to unveil their Astral after we submitted the S10. And so we were like, and then our Lumos died. So we were like, we need a mass spec, and so we bought that. But so I think if you can swing having to, because then you don't have to deal with and when we're using the Astral, we have to deal with putting FAMES on when we're doing single cell, and that it's just a little extra barrier because we actually have to change the position of our needle every time we do it, and then vice versa on the other way around. The Ultra doesn't do well above 25 like you kind of saturate the signal at 25 nanograms. And you don't want to dirty the TIMS cartridge by adding a ton of samples. So
yeah, I feel that for cores like ours, which are not very instrument heavy, we have just one Orbitrap Astral, which works great for all the kind of samples that we throw at it. But what keeps us from optimizing new workflows like SCP, although it's not new. Clearly, you've been working on it for quite a few years. So, but you know what keeps us, the barrier for us to optimize SCP workflows is that we don't really have the time or an extra instrument to start optimizing on, and on the side keep running our core, without a hitch. So so we're trying to find a balance between the two using just one instrument.
No, that's definitely hard, and that was what we were running the issue before we got our second instrument too, where we could just dedicate one entire time to that. So that's when our pace of development really picked up once we got that. So I understand that challenge. Luckily you can run through samples pretty quick, unless you have the problem of your Astral is completely utilized, which wouldn't be a bad problem.
Yeah.
But you can, removing frames is a kind of a pain in the ass.
Okay. So one very little, it's a very technical question.
Yeah.
So when you say you get around 3,000 protein IDs from your EvoSep plus Ultra combination per cell, how many precursors do you get? And versus the number that you get for Astro, which was around 2,000?
So for precursors, we get, on the Ultra, we're getting, I want to say around 20,000 precursors. With the Astral, we get more precursors, but that's because for our low input method on the SP, we're really targeting the +2 ion cloud.
I see.
So the Astral, we're just taking everything that's not +1. So that's why we're getting more I think peptides are more similar counts, but the precursors are different between the two, at least for us.
And that makes a lot of sense, right? So you tune the ion mobility into this really tight region, and so you'll get the total goal is to get less precursors and more proteins, right? So if you start to open up those ion mobility windows because you're interested in acetylated peptides or phosphorylated peptides, and they're just not in that normal ion cloud, you will see the number of the number of protein IDs go down, but the coverage per peptide or the coverage per protein to increase. And to your previous question, my research lab is solely dedicated to single cell. We never run above. I would prefer if the instrument never sees more than 2 nanograms, and so our bulk is 25 cells. If I'm still quantifying 5,000 proteins per cell, I'm really happy with those data. While I was at US HUPO, my team really wanted to see what 50 nanograms looked like on the Ultra, and then we spent 3 days cleaning it, right? So, I don't know that you always have to do that, but the fact that I've got an Ultra that just never sees more than a 10, 10 times the normal cell load, at maybe 10 nanograms of protein at the very tip top, that's part of the reason that was the first time we cleaned it. So it does help, and I know how impossible that is for most facilities, right, to do that kind of thing. But in an ideal world, we'd just be able to download the methods that work from somebody else's lab. You could probably just copy his timsTOF Ultra methods, his EvoSep method, and just not do any optimization on the same system. But we know that's not exactly how it works. But
Thank you.
yeah.
I think we.
Yeah. Yeah. Thank you for excellent talk. I learned a lot. So my question is, if you set up the single cell protein, right, systems, so you list as a thermal arbitrage Astral, also the Brucker time TOF, and this one. If we started to do the protein, single cell protein, so do we need to buy this too?
You kind of talked a little bit about that.
Any instrument could get you single cell proteomics data. It's just how long does that take, right? The Orbitrap Exploris 480 is now an old instrument, but there's really good data out there. Over 1,000 proteins per cell. I've seen that instrument for $400,000 with a decent academic discount or a refurb. So now, is that 10 cells per day? But right, so you get this lower throughput, but is that enough for you to now write your S10, right? Or write, to get one of the instruments you could dedicate to that kind of thing. That's always a possibility, right? But, the most cited single-cell proteomics paper was that's ever been done, right? It's the Scope 2 paper. It's a Q-Exactive Classic. You can get one on eBay for $55,000, like right now, right? You're on your own to fix it, but that data is still really, really good. We were reanalyzing it just this weekend, looking for something. So I think any instrument can do it. If you want to do it really fast, the vendors will take $1 million, and they can give you something that can do it easily and fast, right?
NPNNL is doing a lot of good data with their trigrids.
Oh yeah, no, absolutely.
So
yeah.
if you want to use if you have a trigrid instrument, there's a lot of good. The
Yeah.
benefit of that is you can do the TMT. I don't know what you guys are up to, the 32-plex, which even if you're doing longer methods, you're still then getting at least decent throughput with FAMES and then the MS3.
Oh, James Fulcher, any of the Fulcher et al. Papers, those are really good.
Ben, you said you were trying to move to direct inject, and you think it will affect the quality of your column and the life of your column with so many SPD per day, the separation column, or direct inject?
Yes. So at Hopkins, I had an instrument that was just doing multiplexed TMT. It was a timsTOF Flex with an XR cartridge, and then and I had to, you definitely had to trap on that. And then I had the SCP, one of the first ones out, and I was just doing direct injection on that. The problem was that I was like, that an ion optics column was only lasting a couple of days, right? Because there's lipids from the cells, they're not going anywhere. That's why I think you have to have a trap. And in my mind, I thought, I'll just talk louder. I thought that trapping, having an online trap, would mean that all proteins would just go away.
Yeah.
And Claudia Ortega, when she was at Carl Meschler's group, she showed a really nice trapping protocol where it looked just fine. And then you see the stuff from Jenny Van Eyck and the stuff from Ryan Coe where they're doing the trapping, it's fine. I think if you're direct injecting everything from the cell, the lipids, the DNA, everything into the column, those columns don't last very long, and you can make your instrument alive.
We also do trap when we're doing stuff with the Vanquish. We do trap in a loop.
You do trap in a loop.
Yeah.
Okay, yeah. So we're moving to trap in a loop, and
Yeah.
hopefully we'll trap in a loop. Hopefully soon.
Oh, so that's the future plan, trap and elute for single cell without using EvoStep?
Possibly, yeah.
Okay.
I love the Evo stuff, but it's creeping up in price. They were just acquired, right? And so if I was trying to sell my company to somebody else, I would have the tips really cheap, and I would say, look, you've got 600 users around the world. You can charge them anything, right?
Yeah.
And so I'm suspicious that the tips are going to become $17 fees or something, right, in the next couple of weeks. But they haven't yet, and so we're still using the Evo stuff, but we're preparing multiple. We're currently optimizing dual travel.
Thank you so much.
We ran over. This is
Yeah.
a lot of conversation.
Yeah, I like that. I'm part of that. Okay. If anyone's no more questions, thanks for coming.
Thanks, everybody.